Hello and welcome to the Golden Helix SVS 7.7 in Action webcast. My name is Greta Lindsay Peterson and I am the Product Quality Manager here at Golden Helix. In the past year, we have added exciting new features to SVS and I am looking forward to showing them to you today. If you have any questions, please feel free to enter them in the questions pane of the GoToMeeting dialog at any time. I will address as many questions as possible at the end of the webcast. Any additional questions will be responded to by email or phone call. Today I will demonstrate our enhanced RNA-Seq solution, our streamlined DNA-Seq workflows, and a few other important improvements to SVS. Throughout this webcast, I will be mentioning several features that are discussed in detail in tutorials and the SVS manual. Please feel free to consult these resources for further information and technical details. In particular, the RNA-Seq tutorial on our website has been rewritten for SVS 7.7 and includes information on quality assurance measures and numerical association tests that I will not have time to go into today. As with many of our tutorials, we have a demonstration project available for download, and you can either access the tutorial on the website or download it as a PDF to print or share with others. In addition to having the demo project available, all the BAM files for the samples mentioned in the tutorial are available for streaming from the cloud for visualization in Golden Helix's free genome browser, Genome Browse. The SVS RNA-Seq solution starts by importing tabularized quantification data from the Golden Helix pipeline or other pipelines formatted using the Golden Helix tabularized quantification specifications. One of the most common questions that we have is about how the data should be formatted to be imported into SVS. I will show you how the data needs to be formatted. Once the text file is generated, it can be imported directly using the RNA-Seq Tabularized Quantification tool. Golden Helix can also support output from top hat conflicts. Please contact support@goldenhelix.com for assistance with this data. After import, SVS contains methods for analyzing your data, including principal component analysis, numeric association tests, numeric regression, and the DNA-Seq algorithm. Once the data has been analyzed, the results can be viewed in a Manhattan plot, a heat map with dendrograms, and in Genome Browse, which supports BAM and annotation track visualization. The tabularized quantification format is a very simple format with a really long name that can be tailored to either report counts at a gene or isoform level. The text file should be tab separated. Extension can either be TXT, TSV, etc. and should contain the number of reads mapped to each gene or isoform. The first row should consist of column headers and include in this order ID, chromosome, name, start position of the gene or isoform, stop position. The fifth column can either contain a semicolon separated list of transcripts if the ID is a gene name, or the gene name if the ID is an isoform or transcript name. Samples should start in column 6. And again, the data should be the number of reads mapped to each gene for each sample. Once this text file is created, it can be imported by selecting the RNA-Seq Tabularized Quantification menu item in the SVS import menu. The differential expression analysis method we chose to implement in SVS is the DNA-Seq method from the Anders and Huber paper from 2010. In SVS 7.7, the DNA-Seq dialog has been streamlined and updated to include all the latest recommendations and features in other implementations of the DNA-Seq method. The method in SVS is now consistent with DNA-Seq 1.10.1 available from Bioconductor. Here are a sample of the visualizations that can be created using our tutorial and visualizations that can be created for RNA-Seq quality assurance and with the output of analysis methods. In the upcoming demo, I will show you how to create a dendrogram with heat map in the volcano plot, which is a plot of negative log 10 p-value versus log 2-fold change. The top genes will then be selected from the volcano plot in the BAM files interrogated in Genome Browse to determine if the genes are really differentially expressed or if something else was going on. Before we go into the demo, First, a little information about the samples and the data. 15 breast cancer cells from 3 different cell types were processed using an Illumina HiSeq 2000 and a poly-A selection sample preparation. This ensures that we should be looking at mRNA that codes for proteins. The 3 cell types are basal, claudin, and luminal. This PCA plot was generated from the first 2 principal components after performing PCA analysis on the variance stabilizing transformation that is a normalization and transformation method in the DE-Seq algorithm. We can see that the first 2 principal components are driven by cell type. This is what we would expect for normalized data and indicates that there is not a batch effect with this data that will interfere with detecting differential expression between the genes. The demo will start with the data already imported into SVS in a phenotype file joined to the read counts per gene spreadsheet. Then we will interactively run DE-Seq between the claudin and the luminal groups. From the DE-Seq output, we will generate 2 plots. Of course, you can do all of this yourself using our tutorial. Finally, we will show you our top 2 genes in Genome Browse. Now let's switch over to the SVS project. In this project, I have the breast cancer subtypes joined to the gene counts. I also have some other work that I've done here that we'll start with first. I've transformed the data so that the counts are in columns, or the samples are in columns. And I would like to look at side-by-side box plots of the untransformed counts. By looking at this, I can determine that it would not be wise to work with the untransformed counts because of the variability of the data. In fact, most of this data, you can tell, consists of outliers. Running DE-Seq on untransformed data would not be able to pick up on differentially expressed genes as there is so much variability. After running DE-Seq and outputting the VST, or variant stabilization transformation, on the counts and replotting the box plots, we can see that the medians between all of the samples are fairly consistent. And in fact, there are only 2 outliers. This means that the read counts are not as skewed in this transformed data, and we should be able to very easily pick up on the differentially expressed genes. Now we'll go ahead and open up our spreadsheet. I'm sure most of you are familiar with spreadsheets and SVS, but just a reminder, here we have a categorical column consisting of cell type, and we have the read counts for the genes in columns 4 on. And we have a green map button that contains the chromosome position, which is really the start position of the gene, the stop position, and the list of transcripts. This is all information that was in the Tabularized quantification text file. To run DE-Seq, we go to the Analysis menu and select DE-Seq Analysis. If this option is not available, it is probably because you do not currently have the RNA-Seq package licensed. Feel free to contact your account manager for a demo of this package. Once the DE-Seq dialog is open, we're going to switch our condition A to claudin and our condition B to luminal. Again, we're looking for the differential expression between these 2 cell types. We're also going to select that we want to output counts for the top 50 genes, the VST transform counts. We'll use these counts to create the dendrogram and the heat map. We'll go ahead and click OK. And we're going to scan and look for and calculate the p-values for all of the genes. Once this is done, we will end up with 2 spreadsheets. One spreadsheet containing the p-values and other statistics, and another spreadsheet containing the VST transform counts for the top 50 genes. We will start by looking at the VST transform counts. The first column are the VST transform counts for the most significant gene. The second column is for the second most significant gene, and so on. To create a dendrogram and heat map, we go to the plot menu and select dendrograms and heat map. We have the option of distance metrics of correlation or Euclidean for both the top and the side dendrogram. We also have different options for linkage methods. We will keep the defaults and click OK. And you can see that very quickly we have created a dendrogram and heat map that shows the differential expression between the 2 cell types for the top 50 genes. And the top gene was LEPROL1, and you can see that that's the fourth one down, and there's nice clean separation between the two cell types. Next, we'll create a volcano plot from the p-value spreadsheet. We'll go to Plot, XY scatter plots. Again, the volcano plot is a plot of the log two-fold change on the x-axis and the negative log 10 p-value on the y-axis. Select those two options and hit Plot. And you can see the nice volcano plot. Click on the top gene on the left, and that's the LEPROL1, and the top gene on the right, and that's the GRHL2. Now we'll go over to our History tab. I'm going to double-click on this gene name and copy that. Now I'm going to jump over to Genome Browse. This is a Genome Browse project with all 10 samples loaded into it. These samples are streamed live from the cloud. And at the bottom I have RefSeq genes, but I can also add another gene track if I want. Here I've used our enhanced labeling text editor to add in red an indicator that the first 5 samples are the Claudin samples and the next 5 samples are the luminal samples. Let's go ahead and add that known gene track. So I'm going to click on the Add button. I'm going to stream it from the cloud. I'll just type in known, select known genes, and add to project, and that's going to add it to the top. Now I have in my buffer that gene name, so I'm going to paste LEPROL1 and click Enter. And it's going to go ahead and jump there. Now we can see that there are numerous reads in the pilot view for the Claudin samples and only a few reads for the luminal samples. So there's a clear difference between these two cell types for this particular gene. So this gene is differentially expressed. And the reads for the luminal samples do extend across intron regions and exon regions. Now let's go back to SVS and select this gene name. Jump back to Genome Browse and jump to this one. For this gene, GRHL2, you can see that there are only a few reads for the Claudin samples and numerous reads for the luminal samples. And there does seem to be something interesting going on towards the right side of this gene. So I'm just going to scroll in. It's going to go ahead and scroll in. Now it's calculating the pilot view. Right now I have it set so that mismatches are emphasized. And as I continue to zoom in, there are different variants present in the reads for these samples. So even though these samples are differentially expressed, the variants are different between each sample themselves. And let's actually scroll a little bit to the right some more. We do have some more variants. This region here. Zoom into the very end of the gene, and we can definitely see that there are a lot of interesting variants going on in the UTR3 region of this gene. Now let's go ahead and switch back to our presentation. And we're going to move on to DNA-Seq. In SVS 7.7, we are pleased to announce new tutorials that feature workflows tailored to specific hypotheses and goals. These tutorials include features that have streamlined the workflows to interrogate the data in an efficient manner and are easy to use. In particular, the complete trio analysis workflow, first introduced by Dr. Ken Kaufman in the August 2012 Golden Helix webcast, has been further refined to reduce the time it takes to get to a short list of variants from 2 hours per trio to 20 minutes or less per trio. Later, I will demonstrate some of the new tools for this workflow. The new and updated complete suite of DNA-Seq tutorials include Introduction to DNA-Seq. This is an introductory tutorial covering the basics, including importing data, removing low-quality variants, filtering down to rare non-synonymous variants, and variant classifications. This tutorial is relevant to all workflows. We also have a tutorial called Finding the Causal Variant of a Novel X-linked Disorder. This is the companion tutorial to the October webcast on the analysis to replicate the results found by Dr. Golson-Lyon. Next, we have an NGS family analysis tutorial. This tutorial includes tools to identify de novo mutations compound heterozygous polymorphisms, and rare recessive homozygous polymorphisms. We have a tutorial on rare variant analysis of complex diseases. This tutorial includes workflows for large case control studies, including collapsing methods such as CMC and KVAC. Finally, we have a tutorial on variant filtering on unrelated samples. This includes filtering, variant classification, and tools to find variants unique to affected samples. To access these tutorials, please visit our website and click on Tutorials in the Resources menu. The complete trio analysis workflow uses public data obtained from the 1000 Genomes project. The samples chosen were a trio of Ceph samples, and the BAM files were reprocessed using 7Bridge's secondary analysis pipeline in the latest GATK version. The VCF files were imported into SVS, and then the genotype calls within SVS were filtered down to remove variants with low genotype quality and read depth, leaving only high-quality variant calls. From the filtered dataset, 3 different inheritance patterns are examined to look for potentially putative variants, including rare recessive polymorphisms, compound heterozygous polymorphisms, which I'll refer to as compound hets for short, and de novo mutations. If the child was a male, the variants on the X chromosome could be examined for an X-linked mutation. Other custom mutations can be sought as well using existing tools. As this trio is from the 1000 Genomes project with no phenotype information other than gender and population, for the sake of demonstration, we will treat the child as affected and the parents as unaffected. We will look for rare recessive polymorphisms, compound hets, and de novo mutations. This trio includes a female child, so we will not look for an X-linked mutation. After importing the genotype calls, the read depth, and the genotype quality scores from the VCF files, the first step is to filter the genotype calls to remove any calls with poor read depth and poor genotype quality. The read depth scores are integer values that range from 0 to approximately 250 in this case, although on this histogram I've zoomed in from 0 to 50. The scores from all 3 samples were compiled and one histogram plot was created. After looking at the distribution of read depth, a cutoff of 10 was selected. We will keep all genotype calls for read depth greater than 10. The genotype quality scores are continuous values that range from 0 to 100. The scores from all 3 samples were compiled, and by examining the distribution, a cutoff of 20 was selected. Again, we will keep all calls with genotype quality greater than 20. Once the thresholds for filtering were chosen, the Set Genotypes to No Call based on additional Spreadsheets tool was opened from the Genotype submenu of the Quality Assurance menu. Just a quick reminder, for more information on how to locate this tool and other tools referenced but not demonstrated, please see the SVS manual or one of the DNA-Seq tutorials. Before filtering, the child in a 12878 had 1.46 million variants, 797,000 singletons, 1,143 Mendelian errors. After filtering, the child only had 116,000 variants, 25,000 singletons, and only 11 Mendelian errors. So you can see the filters are successfully removing low-quality variants, which should result in fewer false positives in our downstream analysis. In the demonstration, we will go over the analysis workflows for the following models. Rare recessive, the child inherited one copy of the alternate allele from both parents at the same location. Note these same methods work on indels as well, but for brevity, this will be referred to as an allele. We will assume that both parents donate the same alternate allele. For this model, we will consider mutations rare if the alternate allele frequency is less than 1%. A compound het. The child has a heterozygous call in at least 2 places within the same gene. To be a compound het, an alternate allele has to come unambiguously from the mom at one position and another from the dad at a different location within the same gene. For this model, we will consider only variants with an alternate allele frequency less than 5%. De novo, the child has a heterozygous call at a location where both parents are homozygous for the reference allele. This is a specific case of a Mendelian error. X-linked. Again, this mutation only makes sense for a male child and as such will not be demonstrated. However, SVS makes it really easy to identify locations where the father is homozygous reference and the mother is heterozygous, and the child is hemizygous. Please see the X-link tutorial in the October webcast for more information. Now we will move over to SVS to perform these analyses. In SVS, I already have the data pre-filtered down to high-quality functional variants. I have different starting spreadsheets, one for each analysis that we are going to take. If you notice, I have a green node flag, which is a new feature, in front of each of my 3 starting spreadsheets. There is also a white N on the green node flag, indicating that I have taken some notes as to how I want to proceed with these analyses. First, let's start with the de novo analysis. Open my spreadsheet. Because of my filtering, I have dropped down to 16,000 variants. And you notice that I have a pedigree applied to the spreadsheet. Since these are trio workflows, we do require that you have a pedigree applied to it. For de novo analysis, we have a website script, Find de novo candidate variants. I will click on that, and we'll just run it really quick. And it will only find one variant, one Mendelian error that is consistent with the de novo mutation. And it also created a subset spreadsheet containing the pedigree and that de novo variant. As you can see, both parents are homozygous and the child is heterozygous. We will look at some variants in Genome Browse in a little bit, but we will continue on with the rest of our analyses at the moment. Next, we'll move on to compound het analysis. I will again open up my spreadsheet. Before I proceed, I first need to filter down to rare variants, again using a threshold of 5%. I will then select Filter by Annotation, Filter on Variant Frequency Catalog. I'm going to use the NHLBI ESP 6500 exomes annotation track available from Golden Helix. And I will use the European American minor allele frequency, and again I'm going to set my threshold to 5%. So now I've inactivated 9,679 variants that were present in the NHLBI frequency track. I'm going to go ahead and create a subset spreadsheet. And now I can see that I have 6,700 variants left to examine. I'm going to run quality assurance, score compound heterozygous regions. If you have multiple children in a family, you can get one spreadsheet per trio, or by default, all samples in one spreadsheet. There are other additional outputs that you can get, but we'll just keep the defaults and run. Very quickly, we can score our genes to determine if there is a compound het present or not. I'm going to go ahead and sort total compound het in descending order. And this first column is an indicator column indicating whether or not there is a compound het for this particular trio. Since there's only one trio involved, the maximum value that this can be is 1. If I expand this over, I can see that the total number of inherited hets changes. It's a minimum of 2 and then goes up to 5 for this particular gene. We'll jump out to this gene in Genome Browse. Let's actually go ahead and do that now. I'm going to right-click and copy label and then jump over to my Genome Browse project. As you can see, I have the child, the dad, and the mom in my project, as well as the ref-seq genes. Go ahead and paste in that region, hit enter. And you can see that the child has definitely inherited variants from the dad that do not exist in the mother, and variants in the mother that do not exist in the dad. In particular, let's look at this pink variant here. If we hover over it, it says it's a deletion. That's pretty interesting, so let's zoom in. So I'm just going to scroll in, expand this in the child. So it's a 3-base deletion, and it spans 2 amino acids. So this is definitely a non-synonymous variant. We can go ahead and show the 1,000 genomes variant frequencies track. Now again, this is 1,000 genomes data, and so it's not surprising that this variant is present in the 1,000 genomes database, and it's actually fairly common variant. Scroll back out again. We can see that there are other variants that are present in the child that are also present. Again, not surprisingly, in the 1,000 genomes database. Let's go ahead and jump back to SVS. And we'll go ahead and run our rare recessive analysis. Again, for this analysis, we are going to filter, but this time we are going to use a frequency of 1%. We use the same NHLBI track. We use the default, and OK. And do a column subset. This time I've dropped down to just under 6,000 variants. Now we have a new tool called Activate Variants by Sample Genotypes. This allows you to explicitly specify the inheritance pattern that you are looking for. So again, we're looking for rare recessive polymorphisms, homozygous polymorphisms. So we want the child to be alt-alt and both parents to be alt-ref. Once we choose those options, we'll hit OK. And now we're left with 26 variants left after subtracting the 6 pedigree columns. Create a column subset spreadsheet. And you can see that we have some deletions and some SNVs. But in particular, I want to take a look at this SNV in chromosome 5. I'm going to go ahead and right-click on this column and select Copy Column Header, and then jump back to my Genome Browse project. I'm going to paste that into the genomic location bar, and I do not even have to remove the -SNV. Just hit Enter, and it's going to jump to that variant. I'll go ahead and zoom out just a little bit. And you can definitely see that it's within an exon region, and we'll zoom back in. Shrink this up a bit so we can see all the samples. So you can see that the child is definitely homozygous and both parents are heterozygous at this particular region. And we'll zoom back in. So we can see the coverage a little bit cleaner. Again, homozygous call. In fact, there were no reference calls, and both parents split about halfway between the G and the A for a heterozygous call. And we can again look at the 1,000 genomes track, but this time the variant is not within this track. All right, let's go ahead and switch back to our presentation. We have implemented other DNA-Seq tools within the latest version of SVS. In particular, one of these new features is the ability to build a pedigree from row labels. One of the biggest hassles for our customers working with small families was creating a pedigree file. They knew which of the samples were the mother, father, and child, but they did not have a pedigree file for their data. And it was a hassle to go out to Excel to create this for such a simple pedigree. We listened to our customers and built a tool to build a pedigree based on the row labels of a spreadsheet. All you need to do is to launch the Build Pedigree from Row Labels tool in the Edit menu from a spreadsheet. On the first screen, you just need to specify the family name, identify the father and the mother, and set the affection status, if known, and indicate how the rest of the rows should be handled in the following dialog, as well as if the rows should be sorted in father, mother, children order, or in the previous order from the spreadsheet. In the second dialog, if there are more than 3 rows in the spreadsheet, additional rows will be listed. All you have to do is select the sample that you want to be the child, set the gender, and set the affection status. You can do this if you have more than 3 rows. You can run this multiple times to build multiple pedigrees, or you can also add multiple children to the same pedigree. This makes it a lot easier to just add that simple pedigree information to your existing genotype or spreadsheet of read depth data. Another tool often requested was the ability to visualize the overlap between samples in a Venn diagram. To meet this need, we created the Compare Variance Across Spreadsheets tool in the Tools menu from the Project Navigator. This utility counts the number of shared samples between all combinations of the selected spreadsheets. If 2 or 3 spreadsheets are compared, a Venn diagram is created. Here you can see the number of unfiltered variants for the Ceph trio from the demo. The green circle is the data from the mom, this teal circle is the dad, and the purple circle is the child. Not only does it report the number of variants are in each group, it also reports the number of overall variants that are informative. If more than 3 spreadsheets are selected, a report on the number of shared variants between all combinations of spreadsheets is created. So even if you have 10, 12 groups, you can still get a report on how many variants are shared between all of those spreadsheets. Last but not least, numerous other changes have been made to SVS. I would like to mention in particular our new SVS welcome screen that now features a live feed that lists the top technical support bulletins. Once you update to SVS 7.7, you'll be able to see the release note by clicking directly on the link for that bulletin, and in the future, you'll be able to view the release notes before you update to future releases. This will allow you to decide when and if you want to update, as well as to keep track of all the changes. We also now have an improved Complete Genomics import tool that allows you the choice of how to represent variants that are not called for a particular sample in the same manner as the VCF import tool. You can either choose to fill holes with ref or with missing genotype call. Illumina Final Report Importer has also been completely rewritten to be faster, more computationally efficient, and it also now allows you to import unmapped data or data with a position of 0. Finally, as demonstrated in my projects, we now have the ability to add color flags to nodes in the Project Navigator. The flags indicate whether or not a node has a note attached to it. There are also 4 colors available to flag a node for future reference. This makes it easy to let others know which spreadsheet you used to start your analysis and what was the final result spreadsheet, as well as any other important intermediate spreadsheets or where you left off and need to start your analysis again. I want to thank all of our customers who provided feedback and requested features that went into this release. As I look back on all the new features that have been written in the past 10 months, I am amazed. We will certainly keep the updates coming, and I hope that you will continue to provide the invaluable feedback that helps to make SVS a more comprehensive and robust solution. If you would like to try out the RNA-Seq or DNA-Seq functionality, please contact your account manager or email info@goldenhelix.com. Finally, I would like to encourage you to give Genome Browse a try if you have not already. We have a special offer for the attendees of this webcast. The first 10 webcast attendees who download and register for a Genome Browse account will receive a free t-shirt to go along with Free Genome Browser. It is our Thanksgiving gift to you. If you have any questions and have not already typed them in the question pane, please do so now at this time. Thank you. We have a question, how do I get the de novo candidate script and other add-on scripts? If you go to SVS, open up an SVS project, go to the Resources menu, which is a new menu that we have, and click on Add-on Scripts. It'll jump you directly to the Add-on Scripts page, and you just scroll down, click on Find de novo candidate variants, and download the script there. You will need to enter in your email address, but other than that, it will download, and you can also download the documentation as well. We have a question, what options do you have for importing RNA-Seq data? Again, right now the format that we support directly is importing tabularized quantification data from our Golden Helix pipeline, or you can reformat your output from your own pipeline in the tabularized quantification format that I indicated earlier. We can also support top hat and cufflinks with assistance, so just contact us if you have top hat cufflinks data, and we will help you get it into SVS. We had another question, what else is included in SVS 7.7 and where can I find more info? Again, we have quite a bit of changes in SVS 7.7. You can find more information on our website, or for the particular features that are unique to 7.7, again, from the SVS. If you've already updated to SVS 7.7, you can click on the release notes in the support bulletins, or you can go to goldenhelix.com and go to support, technical support bulletins, and read the release notes. You can also see the release notes from the past several releases to get an idea of what's included in SVS 7.7. One more question. For the normalization of RNA-Seq data, what is supported in SVS? Again, through DE-Seq, we have the VST transformed counts. We also have a normalization and log transformation. So this is an upper quartile normalization, and we also can do a log transformation, and that's within the quality assurance menu. So again, we've got VST, the variant stabilization transformation. There's also a scaled transformation through DE-Seq, and then we also have the normalization and log transformation method. Can you access Genome Browse from SVS 7.7? At this time, Genome Browse is a standalone program, and you cannot directly access Genome Browse from SVS. That will be integrated at some point in the future, but for right now, you do need to have both SVS and Genome Browse, and copy and paste to go back and forth between the two. Do the RNA and DNA-Seq packages work with non-human data? And if so, which species does it support? Both packages will work as long as we have a reference genome, and we do have reference genomes for several non-human species. You can find out more information on our website. You can also, from SVS, go to the Annotation Track Manager, download from Network, and we have a short list of species in the Annotation Track Manager. For instance, for Bos taurus, we have 2 reference sequence tracks, the CBCB or UMD 3.1 track and the older Baylor BTAU4 track. We're continuously updating and adding more reference sequence tracks. So if you do not see the species that you are working with, please contact us and we will add it to our list of species that we're curating data for. I did have a question about demonstrating creating a Venn diagram. It does take a little bit of time to run depending on how many variants you have, but I can show you how we can get that set up. If you don't already have 3 separate spreadsheets, you would want to open your genotype spreadsheet, go to quality assurance, subset informative genotypes by category. Again, since I'm comparing variants between samples, I selected to create 1 spreadsheet per sample, so I'd keep row labels. And once I ran that, I ended up with 3 spreadsheets, 1 for each sample, and I would select those in the variant, compare variants across several spreadsheet tool. And I would hit OK. And again, this takes a little bit of time to run, but in the end, I end up with a variant comparison count spreadsheet and a Venn diagram. And this image can either be copied and pasted in a document or saved as well. What updates are coming in the future for DNA and RNA-Seq? That is to be decided. And again, we are soliciting feedback from our users as to where you would like to see us go in the future for DNA and RNA-Seq. So if you have any suggestions, please let us know. When some of the things that we have talked about are importing other analysis methods for collapsing, methods for DNA-Seq, including SCAT-O or SCAT-U. For RNA-Seq, we've thought about other differential expression analysis methods, but again, that we haven't decided on exactly where we're going in the future. If you have any questions, whether it's about sequence data, working with your sequence data, RNA-Seq data, DNA-Seq data, please email support@goldenhelix.com and we can help you on an individual basis, either by pointing you to the place to go for more information or by setting up an individual training session. How does the RNA-Seq package integrate with my EA pipeline? The output of the EA pipeline is the tabularized quantification output. that can be, again, directly imported into SVS, and quality assurance measures can be performed directly on that, and analysis can be run on that as well. So again, the output of the EA pipeline directly goes into SVS for analysis. Is Genome Browse really free? Will it cost in the future? Yes, it is really free, and no, it will never. You'll never be charged for it in the future. We may have additional applications that are optional that can be added onto it that there might be a charge applied to. We will continue to develop and add new features to Genome Browse, and it will continue to be free for all time. All right, so I think that's all of our questions. Again, if you have any other questions, please email support@goldenhelix.com or contact your account manager. And again, I would encourage you, if you have not already done so, to download and register a Genome Browse account. And thank you all, and have a great week.